Hello everyone, I hope you're doing well. So in this last video in the reproductive system, it's going to be similar but different. So we're going to talk about early embryonic development, look what happens if an oocyte and a sperm become a fertilized egg known as a zygote. What are the stages that lead up to implantation, where the zygote embeds itself in the uterine wall. And then we're going to kind of change topics a little bit and go over a little bit about Mendelian genetics and inheritance and some genetic abnormalities. So you'll be able to learn a little bit about how inheritance of certain genes and traits can occur. All right, so we're going to look at the stages of the embryo. We're going to look at Mendelian genetics, and we will look at a monohybrid and an example of a dihybrid cross. And I'll explain what that means later. And then we'll look at some genetic abnormalities like Down syndrome, Klinefelter's, and Turner's syndrome. All right, early developmental stages here. So when you get to fertilization of two haploid cells, a sperm and an egg, one from the father, one from the mother, you then equal a 2N zygote. So that's just one cell that has both genetic information from the mother and the father in it. Now, that oocyte stays about the same size, and the cells inside begin dividing. They go from 2 cells to 4 cells to 8 cells. So these are the early stages of development. The size of it doesn't change, and no implantation has occurred yet. So these cells just keep dividing. And they get smaller and smaller with each round of division. All the cells are still the same at this point. And then it becomes a mass of cells known as a morula, right here. So 16 cells that are together then. The morula then becomes what's known as a blastocyst, so 70 to 100 cells. And this is roughly within like 10 days after fertilization it takes to get to this point here. The blastocyst then begins opening up a cavity called a blastocele, which has an outer layer of cell known as a trophoblast cells. These cells are what end up becoming the placenta and produce human chorionic gonadotropin for a pregnancy test. The inner cell mass in here is what eventually will become the baby, or the fetus. And so this is just very early on in development. At this point, then, so this is all usually, so fertilization usually occurs in the uterine tubes where it gets to this point. Here, this is in the uterus. Every now and then, an oocyte can be released, and the sperm fertilize the egg out in the outside the uterine tubes, and the implantation can happen in the abdominopelvic cavity, and that's called an ectopic pregnancy. Very, very dangerous if that occurs. All right, so this is also kind of showing what's going on here as well. So we get a fertilized egg here, goes through the 2, 4, 8 cell stage, and then becomes a morula at 16 cells, and then a blastocyst, and a blastocele with the opening in it called a blastocele. And then at that point, you get implantation occurring. So the blastocyst rotates and implants into the endometrium of the uterus. So the whole reason of progesterone production was to maintain that uterine lining, the functionalis layer of the endometrium. So here then, when implantation occurs, boom, it's there. And this is what it looks like when it implants into that. So here, those trophoblast cells, which will end up becoming the placenta, implant into the wall. And then you got the inner mass cells right here. And then you see the endometrium grows around those cells so that when the fetus begins growing, it will be within that endometrium. And it fully secures it to that uterine lining. And then implanted embryo continues to grow. And this is about 7 to 8 weeks after conception right there. And typically implantation occurs on the posterior uterine wall. So that's embryonic development. Now, I know I didn't go into too crazy. And also, I didn't talk about the sexually indifferent stage too much here, which occurs right at 7 to 8 weeks. So you can go into a lot more detail on some of the topics here. But this is just a general overview of what happens post-fertilization. All right, we're doing a little change of thought here. And we're going to talk a little bit about Mendelian genetics. Because with reproduction, you pass things down to offspring. And so here, Mendelian genetics, so it's called Mendelian genetics because of the man who pretty much this is based off of Gregor Mendel. So he was an Austrian monk, and he did a lot of work with pea plants, crossing pea plants, and he noticed these heritable traits that he could cross together, and some were dominant and some were recessive, meaning they hid within. And so we come across these now. We know of these as genes, and the different variations of genes are known as alleles. So you can have a dominant and a recessive form of an allele that are carried on your chromosome. So here, this is called a karyotype. A karyotype is an image, an ordered image of all your chromosomes. So humans have 23 pairs. So, you know, N is equal to 23, so 2n is equal to 46. So 46 chromosomes total here if you count each one. 23 pairs, where pair 23 are the sex chromosomes. So this individual has a Y, it's a smaller chromosome than the X, and so this individual would be a male for this particular karyotype. So karyotypes can be done to do genetic analysis early on in fetal development. So this is just getting what chromosomes look like down. And so when all these little bands here, not that these are genes, but these are tagging certain regions, so you can picture them as maybe a gene. And so there are two variations. One of these chromosomes came from the father, and one of these chromosomes came from the mother. So there could be a particular gene sequence on, say, chromosome 3 here, where the father's form was mutated and the mother's form was working fine. Typically, we refer to dominant forms as a capital letter, and sorry for the dog barking, and recessive forms as a lowercase letter. Garbage man. So dominant and recessive. I actually need to pause. And we're back. Here's the little culprit. The little barking culprit right here. Rub, 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 rub. I'm going to keep him on my lap for the rest. All right. Oh, you're stuck in my shirt. All right. We have dominant and recessive then. Capital letter, lowercase letter. All right, let's get back to my train of thought. So Gregor Mendel, he did this, and he called these, things traits, these dominant and recessive forms. Now we know them as alleles of genes. So, for pea plants, he found that some peas were nice and round, and others became wrinkled. So the round ones were a capital R, and the wrinkled ones were a lowercase r. But then he found these weird things that happened where if you cross two round peas, sometimes you get a wrinkled pea. So how does that happen? Some reason the recessive form hid. And he figured this out by doing these little Punnett squares, is what we call them. And following one gene is known as a monohybrid cross. Following two genes is a dihybrid cross. So you can follow 1 and 2 to kind of see the chances. So a Punnett square tells you what are the odds of producing a certain offspring. So let's say this big A, and little a. So this is known as homozygous dominant, and this is known as homozygous recessive. Heterozygous would be one of each. So these letters represent each of these chromosomes. So up here, big A, little a, this individual would be heterozygous, and you write that as A. So when we write it like that, remember we're talking about chromosomes, and one from each parent. And so when this parent would form gametes, sperm, male, so sperm, some sperm will carry the recessive A, and the other sperm will carry the dominant A. And then it's random which one is then selected and used for fertilization. So that's what a Punnett square represents. It presents the chances of the gametes that could form from that parent, and then from each parent. So here, one parent was recessive, one parent was dominant, and then every offspring then would be heterozygous in this cross. When you do these crosses, you take this row and then this column, and you put them together, and we'll see the difference a little better. But then let's say you now cross this one with its sibling. In a way, it's called a generation 2 or F2 cross. So here you're crossing two heterozygotes. When you cross two heterozygotes now, so you now can see, you have homozygous dominant, you have two heterozygotes now, all of these would all, if we're doing round and wrinkled, all of these, since they have the dominant allele, there'd be 3 round. And then down here, you would then have 2 recessive, 2 little a's, meaning 1 wrinkled. So you'd get a 3-to-1 ratio of round to wrinkled peas. I know we're talking about peas, but that kind of just summarizes it a little better for you. So you can do all these sorts of crosses. So let's say we have a let's say we have a recessive individual, and you cross it with a heterozygous parent. So you could do this cross. So these ones can go across the top, and these ones you go down like this. And then when you do this cross, you draw out your Punnett square, and then you fill it in. So here you would see there's a 50% chance then of having a recessive form and a 50% chance of having a heterozygous dominant form. So that's just how a monohybrid cross, you can see one gene moving through. Another thing we can talk about is a dihybrid cross. Let's say we want to follow 2 things. We want to follow, round and wrinkled, and peas can also be green or yellow. So here you got to define your letters. I'm just going to use simple letters now, A's and B's. So a dihybrid cross is saying, let's say we have a round green pea, green is dominant, and we cross it with a wrinkled yellow pea. So something like that. So a heterozygous and homozygous recessive right here. So this is the same type of cross as that, but now we're following 2 traits. So we have a known heterozygous round green pea, which is the dominant forms of each, and then we have a known recessive one, which is wrinkled and yellow. I know I'm just using A's and B's. So when you form this gametes, then we got to think about random gamete formation. Remember, these are on chromosomes, and this is why I have this up here. Let's say the A's are on this one, and the B's are on this one. When you form these gametes, this A and this B could be together, and then you would have, A, B, like that. Or this A could be with this one when that gamete forms, and that one would be little a, little b. Or you could do big A, big B, or big A, little b. So you have 4 possible gametes that can form, so 4 different gametes that could form. So here, when you write this out, I always do it as first, so FOIL method. First, outside, inside, last. So you get A, B, big A, little b, and then little a, then we could do, so first outside would then be, this one should be big A. So first, and then big A, little b, and then little a, big B, and then little a, little b. These ones here would all be the same going across because there's no differences between them. And then, so when you do a dihybrid cross, you then have 16 possible combinations you can make from all of these. And so you draw this out like this, and then this will then form the dihybrid cross. And when you do that, you would then you combine them then. And you only have to do 1 row here. And you can see you're combining that with that. And so little a, big B. Always write the dominant one first because it's easier to read then. Then here you would have an all recessive. And then all of these would be the same if you would do those. So you don't have to do them all because all these letters are the same. You know they're the same. So what are the different variations we have? So we have phenotype versus genotype. Genotype are the genes. Phenotype is what you see. So this one here, this one would be round and green. This one would be round and yellow. This one would be wrinkled and green. And this one would be wrinkled and yellow. So you would get a 1-to-1 to 1-to-1 ratio of all the phenotypes. So that's just an example of how you can see the chances of different variations showing up by following 2 traits. So 2 traits is a dihybrid, a single trait is a monohybrid. And so if you want to do like simple genetics, like, looking at things being passed in the family and things like that, you can look at simple crosses like this. Now, it does get more complex if, you can get different. Not everything follows simple Mendelian inheritance. And then with this, you can have genetic disorders. So let's move this over to here. Sorry, got to be slid over there. So we have genetic disorders. So disorders can be dominant, recessive, or sex-linked. So you can have a dominantly inherited disorder where the dominant form of that allele is the disorder. Polydactylism, where multiple fingers or toes, is actually a dominantly inherited disorder. Achondroplasia, which is a form of dwarfism, is a dominantly inherited one. Most disorders are recessive because it's rare for a dominantly inherited disorder to be able to be passed down in generations, because typically it could affect the ability to have kids. So recessive disorders are more common because they can hide in a population. And another thing like that with that are sex-linked disorders. Sex-linked disorders are found on the X or the Y chromosome. Typically, when we talk about sex-linked disorders, they're on the X chromosome, which makes them more common in males because they only have one X chromosome. So here, if we're looking at a cross between, a male that has a disorder and the mother does not, you look at the offspring. This is this one's showing an X-linked dominant. So if the offspring has it, they are affected. So the father gave this X chromosome to the daughter, and so all daughters would have it in this case, whereas the mother is giving the X chromosome to the son, so none of the sons would have it. That situation is a little bit more rare. Most are X-linked recessive. So here, if the mother has it, it varies. So there's a 50% chance of whether it's son or daughter. But now let's say it's X-linked recessive, and I'll just draw that up here above my head. So if it's X-linked recessive, you can do X little a, X big A. So there would be the recessive one, whereas the male would be dominant in this. Let's say this cross happened, and let's say this was hemophilia. When you draw out this Punnett square, so there's the mother and there's the father, the Y doesn't have anything. This daughter would be a carrier. This daughter would be homozygous dominant. So neither of these would display the phenotype. However, this son would then have it, and then this son would not have it. So there's a 25% chance of any offspring having it. And if it's a son, there's a 50% chance of hemophilia going, where neither parent shows it, but the mother is a carrier. So that's an X-linked recessive disorder. All right. So now with that, we can talk about some chromosomal disorders that exist as well. We talked about non-disjunction in the last video. So this is a non-disjunction event that occurs in meiosis, where there's typically an unequal separation of chromosomes. Three common ones are Down syndrome, which is also known as trisomy 21, Klinefelter syndrome, and Turner's syndrome. Typically, when you have a chromosomal disorder that results in an unequal number of chromosomes, the individual never survived. The fetus doesn't survive till birth or dies shortly after birth. Chromosomal abnormalities are very difficult to overcome, so that's why typically most of them are on the sex chromosomes. So there's only a couple viable ones that are outside the sex chromosomes. So here, we're showing that karyotype. So for Down syndrome and trisomy 21, there'd be 3 number 21 chromosomes. There could be another weird thing that occurs and make it heritable as well, but 3 number 21 chromosomes, and that leads to cognitive development delays and things like that, and death earlier in life. Then Klinefelter syndrome is XXY. So this individual will have another X chromosome. So XXY in that case. So they develop as a male, but they have more female phenotypes like wider hips, breast development, and things like that. And then you can have other things like poly-X syndrome, where there could be multiple X chromosomes in a female. So there are some other variations. That's not called Klinefelter's, though. That's called poly-X. And then Turner syndrome is where there's only an X chromosome, there's no Y. And so that's written as an O or XO, and that is the only viable monosomy. So a monosomy is where there's only one of the parent or pair. So here, only the father gave an X chromosome, or it could have been the mother, and the other X chromosome never arrived, so there was a missing one here. If number 17 was missing one from the mother or father, this individual would not survive. So when I say it's the only viable monosomy, it's the only one where you can live without one of the sets of pairs. So those are just a couple. There are tons and tons of chromosomal disorders and ways they can happen. You can get different rearrangements. You can get these part switching, and it can get complex. You can take genetics if you want to get into all of that. But this was a little just summary of Mendelian genetics, how inheritance works. So this is how, you can figure out what can pass down in a family and whatnot, and how you can look at single traits being passed, so single variations or alleles of a gene, and seeing how they could pass to offspring, following two at the same time and looking at the chances in the offspring. And then we started out with a little bit about embryology. But thank you for listening. This is the last video of this term. If you made it this far, great job. If you send me an email that says hi, I'll give you some extra credit. And that means you got to the end of the videos and you made it until the end, and I thank you for watching them. All right, I hope you have a great rest of your day, rest of your life. If you ever need anything, please reach out and let me know. All right, have a good one. Bye-bye.